Welcome back, everyone, to the Dairy Science Digest. This is a podcast designed to bring the Journal of Dairy Science straight to the ears of dairy producers. My name is Reagan Bluel, and I'm from the University of Missouri Dairy Team, and today's guest is Dr. Rodrigo Ibanez with the Center of Dairy Research at the University of Wisconsin in Madison. And today we're talking food science. And for those of you that listen in on a monthly basis, you might be wondering, because this is an unusual topic for us, but guys, the dairy proteins that you generate every day is profound. And he and his team are working together to make sure we best understand what's happening molecularly when we process those proteins. So by now, you've all probably learned a little bit about A2 milk, but please listen in today for a deep dive discussion about how this tiny substitution in one amino acid has ripple effects on cheese production. The featured article is a result of industry-led questions needing scientific answers, and this team in Wisconsin wanted to best understand how that proteolysis of milk changes while making cheddar with A1 or A2 milks. So, welcome to the Dairy Science Digest. Thank you so much for joining us today. Could you please take a brief moment just to introduce yourself to the audience?
Yes, absolutely. Hello, everyone. Well, as I was introduced before, my name is Rodrigo Ibanez. I work as a scientist at the Center for Dairy Research, specifically on cheese research, where we evaluate different topics about cheese quality, how we can improve the production of cheese, how we can improve certain defects on cheeses. And as it was mentioned before, we also wanted to see what was the impact of subcases from the genetics of animals on the quality of cheese. That's why we were intrigued by studying the impact of A1, A2 milks on the quality of cheeses.
Awesome. There's several farm families across the nation that have kind of tiptoed into processing their own cheese and milk. And so that, in addition to the industry questions that are doing millions of gallons of dairy production, we just got to understand this better. So if we could please take a brief moment to talk a little bit about the functional properties of milk, and breaking down the caseins are one of the proteins in dairy products. And if we zoom in on that, can you describe the different caseins that are present that you were really wanting to investigate in this project?
What happens is that dairy cows, they can produce, depending on their genetics, they can produce different type of proteins. And depending the types of protein that they can produce based on their genetics, the milk could be more suitable or less suitable for certain cheese making applications.
Mm-hmm.
When we are typically making cheese, we need to form a gel.
Mm-hmm.
Okay, and this gel should have certain characteristics in terms of firmness before we start cutting it and before we start removing the water fraction of milk.
Mm-hmm.
If the milk doesn't have the proper consistency, the proper firmness, we learn that from experience. The cheesemakers, they use their senses to determine at what point they have to cut the gel. Okay?
Yes.
Scientifically, we have some instruments that can help us out to determine what's the firmness.
Yeah.
Based on the genetic of the animal, we may have dairy cows that they produce certain types of proteins that can produce milks that will produce gels with different levels of firmness. Cheesemakers can modulate the when to cut the gel to have the consistency in firmness and therefore the consistency in the cheese composition. So one thing that happens, especially in states like Wisconsin, the cheese making process is done by the time. So if you're making cheese out of A2 milks, you need extra time to cut the gel. At the end of the day, you're losing time and therefore efficiency on the cheese making. Right. Because here industrially, big companies, they make 20 vats of milk per day. So if you're
2 minutes matters. 2 minutes
yes,
times 2 minutes
2 minutes,
times
five
two.
minutes,
Yes, right.
it's
It
gonna matter
matters.
at the end of the day.
Long
And
time.
therefore
Yes.
the efficiency of cheesemaker might be
Hmm.
decreased a little bit. That's from
Yes.
the cheesemaking perspective.
Yes. And guys, what I think is so cool, we've known about this A2 variant since 1961, right? And it's really, in my opinion, professionally, it's really taken steam in maybe the last, I don't know, five years or so, it seems like, or maybe even longer. But there's just this interest from consumers about A2 milk. I couldn't help but feature your article because guys are asking questions about strategically breeding unique sires to meet the demands of what consumers are asking for. So, what did you find in the cheese vat as the concentration of A2 milk is increasing? How is that changing the cheese industry?
Yes, very interesting question. So it's been a lot of interest on focusing specifically in one of the milk proteins that's called beta-casein.
Mm-hmm.
Okay. Dairy cows tend to have either genetic variants, A1 or A2. Okay.
Mm-hmm.
I'm just talking here about one specific protein. When we're talking about caseins, there are four caseins:
Yeah.
alpha casein, beta casein, and kappa casein. And among the alpha caseins, we also have alpha S1 and
Uh-huh.
alpha S2. Each dairy cow has genotypes for each individual enzyme.
Guys, just pause for just a minute. Guys, think about that. They're zooming in so much on just one element of one part of protein that's occurring in your milk, right? You're zooming in, and there's three others that could be, but all of this science, all of these years have really narrowed down to that one genotype, right? And that's what this whole paper is focused around. But I just find that so fascinating. And I'm going to geek out and bring up several different topics throughout your paper here as we talk a little bit more because wow, food science is really cool. And honestly, guys, he's talking about cutting curds as an art. You know, your cheesemakers out there, they can feel, they can see, they can know when to cut these things. Just like you can feel and you can see and you can observe your dairy herd and acknowledge when cows are right or wrong, you know? But learning more about all of this helps you make more educated decisions. And that's what we're doing here today is just educating ourselves about these proteins within your cow herd. Sorry to interrupt.
Oh, no, no,
I
no,
think
no.
that beast is so beautiful. And the fact that we have milk here is what's moved humanity forward all these years. But circling back on the bigger scale of things, you made mention that as the A1 versus A2 are making these different peptides, it changes the yield of that total cheese production because of the proteins being lost to the whey function. I just want to pick your brain a little bit. How do you see the dairy industry changing, or where is it going to move towards as far as, are we just going to integrate this A2 milk into the A1 milk and the cheese industry is going to just accept the reduced yields? Or is there ever going to be a time where we'll start to segregate these two different types of milk?
Yeah, that's a very interesting question, and thanks for asking it. Something that's very interesting from the cheesemaking perspective
Hmm.
is that we can modify the manufacture protocols to avoid losses on the yield.
Oh, well, that's a game changer.
And that's why I was mentioning before that for cheesemakers, it is relevant to determine the firmness of the gel before cutting it.
Yeah,
So they have become experts on that, you know.
sure.
Um, and they have to cut at consistent firmness to achieve the desirable yield. And that way they can avoid
Oh,
the losses of solids.
I see.
So
Awesome.
when we're talking about the A2 milk that they can produce weaker drainage gels,
Mm-hmm.
the cheesemaker can wait a couple of minutes extra
Incorrect.
to
Okay.
correct that.
Oh, brilliant. Good.
Also, during the cheese making process, we have to stir the curd to
Mm-hmm.
remove the water.
Mm-hmm.
If the gel at that point is too weak, cheesemakers can stir
differently.
gently to
Yeah.
avoid that the curd particles will break down and then will be lost in the way. So we have
I can
way,
hear all of those cheesemakers out there listening and they're like, duh, Reagan. So,
and that's,
but no, that's awesome information because this kind of stuff is not inherently obvious to boots on the ground dairymen, right? And I
yeah.
think educating ourselves about this is really important, guys. And so, um, we're making genetic decisions at the farm level, and I think a lot of guys are moving towards some A2 sires, believing that they're helping the greater good, that fluid milk's going to be more digestible for the consumers nationally, and so they're choosing these sires, hoping that long term, we're in it for the long game. Long term, more Americans will be able to drink fluid milk. Is that a reason to adopt these sires? Do you think that will help our industry 20 years from now?
Uh, this is the first step that we want to take by doing this study is seeing the presence of these compounds that may or may not have an impact on human health.
Mm-hmm.
But we wanted to understand a little bit more the proteolysis side of things.
Mm-hmm.
We may be able to modulate the proteolysis in cheese to ensure that these compounds are degraded.
Right.
Is it something that could happen? At this point, we don't know,
Uh-huh.
but further research will be required for that.
Yeah, that's
So at the end of the day, we have to think that we're making food products that not only will help from the nutritional perspective, but can
mhm.
also help on the health of consumers.
Yeah, so I think within that kappa casein and the beta caseins, really zooming in on that, let's talk about your experimental design a little bit.
Oh, yes,
Yes,
absolutely.
you had 26 cheeses that you commercially bought, and they were kind of broken out into two categories. For ease of articulating, you had your A1s and you had your A2s. And I know the A1s included your A1 A2s, but they had A1 in them. And there were 16 of those cheeses, and there were 10 commercially available A2 homozygous exclusive cheese. And those were your 26 experimental units that you were determining these protein differences and how they perform. So let's zoom in and talk a little bit about some of your findings, acknowledging that both protein and fat impacts the quality of the cheese.
Yes, correct. So we got a lot of interest on how cheeses made with A1 or A2 beta caseins could have an impact on the final quality of the product. During cheese making, we're focused on determining the firmness of the gel.
Mm-hmm.
Typically, milks that contain beta-casein A2 tend to produce a weaker gel. If we
Mm,
produce a weaker gel, a lot of the milk solids will be lost in the whey fraction, and therefore
mm,
the yield could be lower.
mm-hmm.
That's one reason that we wanted to investigate. The second one is that after making the cheese, cheeses undergo through a ripening process. A ripening process is a storage time at certain temperature that will allow us the development of the desirable texture and flavor for one specific cheese. The ripening, depending on the cheese variety, can go anywhere from a couple of days up to years.
Well, let me read. Can I quote your article here a little bit?
Sure.
Proteolytic degradation of caseins during the cheese manufacturing and ripening leads to the release of peptides. And these peptides are subsequently broken down into amino acids by microbial activity. And those amino acids are then further catabolized into flavor compounds. A total of 19 individual fatty acids were identified in these commercial cheeses. And so that sentence was so cool to me, talking about how this is almost like a living, breathing block of cheese that's evolving and changing through this ripening process. Science is happening as it's sitting there on the shelf. And what's happening there is creating the flavors that we desire so much. And we know that cheese is one of the top dairy products out there, and that's what makes it so exciting to learn more.
Yeah, absolutely. And if we're talking about flavor development of cheese, the amount of flavor compounds that can be found
Yeah,
when we're doing research is actually overwhelming. We have instruments that can allow us to identify these aromatic compounds.
yeah.
We have found hundreds of compounds that are
Wow.
being produced, and they're really overwhelming to investigate because we don't know
Where do
how
you start?
where to
Yeah.
start. But in the case here that we wanted to also see in this study, and it's something relevant to mention about, is that even though there's only a slight modification in the amino acid composition between the A1 or A2
Mhm.
beta-casein, milk or cheeses that are made using beta-casein A2, they are
Mhm.
during the ripening process, meaning the breakdown of proteins to produce the desirable texture, to produce the desirable flavor development, they also produce other compounds that are called peptides. Peptides
Mhm.
are small protein fraction comprised of small amino acid units, and beta-casein A1 tend to produce certain types of peptides that exhibit what is called an opioid activity.
Mm.
These opioid-like peptides to cause discomfort in certain types of consumers. It's not
Uh-huh.
lactose intolerance. Cheeses typically, and cheddar cheeses typically don't have lactose 2 weeks after manufacture because
Mhm.
the lactose is
It's
fermented.
just gone. It's
Yeah,
used.
so
Yes.
there are consumers that have claimed that after consuming certain types of cheeses will have discomfort.
Mm-hmm.
That discomfort is associated with the release of certain peptides that with this opioid-like activity. And these peptides are being produced from beta-casein, and those ones are called the beta-casomorphins.
Mm-hmm.
So it's a group of peptides that is associated with that discomfort.
Those BCMs in your article was talking about opioid production from the cheese side of things, and I never ever thought that there would be that coming from the cheese. And but that paper in 2024 identified at least 26 different opioid peptides known as these BCMs that are commonly released through enzymatic hydrolysis or during that cheese ripening process. And they're only coming from the A1 cows, is that right?
Well, that's the most interesting part. It comes from both groups, from beta-casein A1 as well as
Uh-huh.
beta-casein A2. So we wanted to investigate what was the occurrence of these compounds,
Mm-hmm.
commercially available cheeses
Mm-hmm.
made out of A1 or A2 beta-casein.
Amazing. And so as we zoom in, I always like to draw attention to the actual paper. Listeners, that there's a clickable link there in the show notes that you can pull the PDF version of this paper up. Figure 1 is very colorful on page 7, and it's got an example of the capillary electrophoresis between an A1 variety versus an A2 cheese sample. And you can visually see spike differences throughout the analysis here. Some of them were statistically significant, some of them were not. And I think kind of rewinding a little bit, in the 26 cheeses that you studied for this particular project, there wasn't there wasn't a statistical difference between the BCMs or these opioid peptides. And so, zooming in a little bit on the fat side of things, I did find it really interesting talking about the firmness of the cheese, and as it aged, it was more likely to crumble if it was an A1 product versus A2. Did I remember that
Oh
correctly?
yes, there are certain, yeah, there is a big difference from the degradation standpoint.
Mm-hmm.
Beta-casein A2
Mm-hmm.
tends to degrade slower than the beta-casein A1. Therefore, if the beta-casein A2 degrades slower, we need protein breakdown
Mm-hmm.
to soften the cheese during ripening.
Mm.
Therefore, cheeses made out of A2 milk tend to be a little bit firmer
Mm.
than cheeses made out of exclusively A1 milk. The same
Okay.
happened with the digestibility, and that's another claim. During gastrointestinal digestion, beta-casein A2 tends to digest slower.
Mm.
And because of that, there's also this claim that they produce less amount of the beta-casomorphin.
Huh. Interesting.
Whereas,
And prior to opening your paper, I had never heard of the BCMs, right, whatsoever. And I didn't understand that there were any opioid peptides whatsoever in cheese. And the first place my brain went was, is that why cheese is so addictive?
that's
Is
that's
that
what
a
been one of the
thing?
claiming. Yeah, that's been
Yes.
one of the claiming. Yeah, but some cases opioid-like products like bioactive peptides with opioid
Uh-huh.
activity, they can cause discomfort.
Sure. And so, what differences did you find between these cheese products that were analyzed?
Oh yeah, absolutely. So we can test milk proteins by performing electrophoresis analysis.
Mm-hmm.
When we have been able to quantify and identify proteins, but also peptide fractions using
Mm.
capillary electrophoresis. We did this specific capillary electrophoresis test to confirm that the cheeses that we purchased, that they were claimed either as A2 cheeses or regular cheeses that had the phenotype A1, A2, were actually made with the specific beta casein it was claimed for.
Mm-hmm.
But we wanted to confirm by this testing that
Mm-hmm.
all the cheeses A2 were in fact A2. But in addition to that, we did a more complex analysis to determine if these beta-casomorphin compounds were found in the cheese. The beta-casomorphins are being released either during cheese ripening, because
Mm-hmm.
during cheese ripening, as I was mentioning before, there is a proteolysis process taking place in which the proteins are breaking down into smaller peptide units
Mm-hmm.
into final amino acids. So in a cheese, we can find this beta-casomorphin. In addition to that, after we consume the cheese,
Mm-hmm.
gastrointestinal digestion can further release this beta-casomorphin. Although we did not study that aspect of
Sure.
the
Sure.
release of the beta-casomorphin, we wanted to see the presence of this beta-casomorphin peptide in our cheeses.
So, if there's a listener here that believes they can't have dairy products, is it a safe statement to say that even your A1 cheese, as it ages, would have less concentration of the opioid that will likely have those undesirable impacts? Is that a safe statement or?
I'm not certain because there are two things. So first of all, in this study, we found these beta-casomorphin, they have different sizes. So they go from beta-casomorphin 3, because it's comprised of 3 amino acids,
Mm,
beta-casomorphin 4, 4 amino acids, and so on.
mm-hmm,
The most common one is the beta-casomorphin 7.
mm-hmm,
Beta-casomorphin 7 has been claimed that is not produced by beta-casein A2.
mm,
Okay? And that has been associated with this reduced discomfort if I
mm-hmm.
consume an A2 milk.
I see, I see. Got it.
Okay. Whereas beta-casein A1 can release the beta-casomorphin cell. And that's just because of this little difference between the amino acid composition.
So when you were analyzing the 16 versus the 10 samples, did you find a difference in BCM7?
Yes, we did find that cheeses that were made out of A2 milks, they did not contain the beta-casomorphin 7.
Hmm.
Okay, that was an interesting finding.
Mm-hmm.
However, we did find precursors of beta-casomorphin 7.
Mm.
Okay, we
Mm-hmm.
found large beta-casomorphins in A2 cheeses
Sure.
as well as in A1 cheeses.
Mm-hmm.
Okay,
Right.
so, and in addition to that, we found other beta-casomorphins, such as beta-casomorphin 9, in
Mm-hmm.
both group of cheeses. Okay, but it is interesting to highlight that the A2 milk since also contain beta-casomorphin 9, that beta-casomorphin 9 is also associated with
Discomfort.
opioid-like activity.
Yeah. Uh-huh.
But more interestingly, I don't know, in the last 10, 15 years, there's been a lot of research taking place on how these bioactive peptides can have an impact on human health. Okay. Beta-casomorphin is just a group of these bioactive peptides. We have done here at the Center for Dairy Research at CDR studies where we're trying to find bioactive peptides with anti-hypertensive properties, with antimicrobial activities, with antioxidant activities, and so on. Many activities that may have a positive impact in human health. When we have tested exclusively in the beta-casomorphins, we have noticed that these peptides may have one or more functional properties, meaning that, for instance, beta-casomorphin 7, it has an opioid activity, but the literature has also shown that beta-casomorphin 7 has an antihypertensive, can reduce blood pressure.
Oh, wow. Okay.
So
Wow.
all the studies that are done, you're not necessarily narrowing everything down to just one peptide.
Right.
When you're consuming a dairy product, hundreds of bioactive peptides are being released.
Isn't it incredible?
Hundreds.
Yes. Really incredible. Well, I can't lie. I knew the BCMs were part of the story, but I didn't understand quite how deep.
Yeah.
And I really appreciate you illuminating that for me a little bit. So my favorite question to ask, and this one might be trickier for you than most, but what's one take-home message that you want dairy producers to learn from this podcast today?
Oh, yeah. Yeah, absolutely. I will say that there is market for any possibility. There could be some dairy farmers that are really interested in these subjects. There will be a group of consumers that are willing to pay the extra dollar for that. And at the end of the day is that regardless, what we're trying to aim for is producing high quality products. And it can be done with any type of milk. And we're trying to understand better how these systems work to produce high quality products. We still need to have a better understanding how these beta-casomorphins can impact on human health. We have seen from other research that these opioid peptides are not only produced by beta-caseins, they can be produced from other caseins and even whey proteins. So we still have to understand how the sum of all of these bioactive peptides interact.
You know, even beyond dairy, it makes me wonder, how does the rest of the diet that the consumer's consuming, how does that play a role for or against their ability to experience these opioid proteins differently? I don't know. I'm not sure. But what I do know is that, Rodrigo, this has been a great discussion. It's been so incredibly fascinating to learn more about the food science side of what we do every day. And I thank you so much for your time and your willingness to do a deep dive into cheese production and even speculate with us about the future of our industry. And I know that this has helped producers really better understand how their breeding decisions may or may not impact the cheese industry moving forward. And listeners, I applaud you for taking time out of your day today to listen in, to kind of tear down some of those silos between milk production and milk processing, and better understand each intricate detail of the milk that you're producing. This is not just white stuff flowing down the road. You are generating a delicious, nutritious product that has so many bioactive compounds in it that we can't even begin to wrap our head around. And scientists just like this man is helping you and all of us best understand how it's impacting us. This has been the May edition of the Dairy Science Digest, a monthly podcast project designed to bring the Journal of Dairy Science straight to your ears. We highlight peer-reviewed research articles because it's sound science that you can base your management decisions around, provided to you by your University of Missouri dairy team. So please be sure to like, share, and subscribe to get future editions straight to your cell phone. This is Reagan Bluel with the Dairy Science Digest, and I hope you have a great